So my name is Steve. I'm a postdoc working with Angela Gronenborn at the University of Pittsburgh. We are just now dipping our toes into the world of the minimal cell. We are a structural biology lab. We primarily use NMR to study proteins, protein structure, protein interactions. Dr. Gronenborn is very well known in the NMR field. She's developed a lot of the methodologies that every protein NMR lab uses. So we think we're well positioned to work with a lot of these proteins and hopefully make some cool discoveries to share with everybody. This is a more aspirational talk at this point. Like I said, we're just diving in the last couple months here. So as John mentioned at the beginning, we're down to about 66 genes of unknown function that are essential for growth in SYN3. So we are focused on looking at these essential genes that are unannotated or have some generic annotations to see if maybe we can figure out what's going on with these proteins. So being a structural biology lab, we approach everything through the lens of structural biology. So of course, we're going to start with a lot of structural predictions. We're going to look through bioinformatics and try to figure out, can we look at a predicted structure and from there start to figure out maybe what these proteins are doing? Or are there proteins that, while not identical, might be similar enough that we can start getting an idea, since a lot of these proteins have no annotated function. And we specifically are focusing on proteins less than about 250 amino acids. NMR has a relative upper limit on size, so we don't want to go too big, especially early on with this. So one thing that caught our eye as we were working through some structure prediction work was this SYN3 0873, I'll just call it 873, 66 amino acids long, so small single domain protein that is quasi-essential in SYN3. The BLAST search tells us that this is highly conserved in mycoplasma and appears to be present in other firmicutes as well. But what was interesting is that our structure prediction returned what appears to be an SH3 or an SH3-like domain. For anyone who has done NMR, that is one of our kind of standard structures that we work with. NMR folks see an SH3 domain and we get really excited. A lot of development work has been done with SH3 domains or SH3-like domains. They're named for the Src homology 3 domain from protein tyrosine kinases. These domains are present across nature. The SH3 specifically is in eukaryotes, SH3-like in prokaryotes. The domain is most often involved in cell signaling or complex formation, particularly through binding to protein-rich motifs or other hydrophobic regions. However, we don't see free-floating SH3 domains in nature, to our knowledge, and I'm happy to be corrected by anyone who has knowledge of this, but in our experience, an SH3 domain is always part of a larger protein. It might be a single domain of that protein, but it's never, to our knowledge, floating around in solution on its own. Even the SH3-like domains from bacteria, they're most often involved in cell wall trafficking and interactions. So that really is interesting that this appears to be a free-floating SH3 in a cell that does not have a cell wall. In discussions with John, he kind of confirms that this truly is a free-floating gene, free-floating protein. It's not formed from some kind of a truncation event. So this unusual single domain SH3 kind of caught our interest, even though it's not an essential protein necessarily. We are interested in using this as a test bed for further method development. So of course, we're going to express and purify this out of E. coli. It expresses really easily, which is nice. One of the great things about SH3 domains is they tend to express really well. They tend to be very, very stable. So that makes it an excellent candidate for NMR study. So we can grow our E. coli in a minimal media supplemented with 15N ammonium chloride as the sole source of nitrogen, and we can purify our protein out of there. And of course, the first experiment that we are going to run is this proton nitrogen HSQC. This is a fingerprinting experiment. Every signal in the spectrum corresponds to one amide group in the protein. Primarily in the center of the spectrum, we're going to see our backbone amide groups, the NHs, and then we have the side chain NH groups up here. But what we immediately notice from this is the signals are dispersed over about 3 ppm. This immediately tells us that this protein is probably well folded. We don't see a huge number of less intense signals, so we're probably looking at a single conformation. To say nothing about dynamics, but we're not looking at multiple population states or probably different folds. And we see, basically one signal for every amino acid in here. So that's really great. This is a prime candidate for us to continue NMR studies. So these are spectra that were just obtained very recently. So again, much more to do moving forward as we try to solve the structure of 873. Is this actually an SH3-like protein? Again, we're very interested to see if this is an example of a free-floating SH3. That would really surprise us that this exists, and we're very curious to see what happens. And we're curious about really what does this protein do. So we're going to try to use some pull-down assays from the SYN3 lysate and try to see if we can pull out some interaction partners and identify those. And from there, ideally, we would be able to do some in-cell NMR. This is where we will prepare our protein in E. coli and then transport it into the minimal cell, and we can then grow this out and observe the protein in its native environment inside the cell. Sorry about that, getting some strange feedback. And then we can see how this protein interacts inside the cell in its physiological environment, and hopefully explore more about its dynamics, whether or not it's involved in forming larger complexes, how the different growth phases may or may not affect what it's involved in. And then, of course, since this is a very small protein, we don't really know what it does yet, but we're excited to extend any of these applications to much larger proteins. So if we, very quickly look through, right, this is a brief list of some of the unannotated or generic annotations with kind of the NMR available sizes all highlighted, right? So we're excited about using something like 873 as a test bed for some method development, and then expanding out and getting into these larger proteins, as well as utilizing other structural methods to both solve structure and use that to guide a search for a function of all of these different genes. So with that, sorry there's not more data. I'm hoping to have a lot more to present next year, so we can hopefully get excited about that. I want to thank Dr. Gronenborn. Obviously, she's my supervisor. Dr. Glass has been very helpful, answering emails, giving us some tips and tricks and pointers, and has been a really nice sounding board for some of our ideas as well. And of course, want to thank National Science Foundation for funding my work specifically. And with that, I'm happy to take questions. Go ahead and just shout it out because I can't see the video view. So go ahead and just start yelling if you have questions.
So Steve, I'm just thrilled that we're bringing new technology to our repertoire of tools to look at things. Have you got any thoughts? You want to elaborate a little bit about what you vague thoughts, precise thoughts about what 873 might be doing in the cell?
Mm-hmm. Yeah, so in some discussion with you, you've suggested that the protein, we haven't been able to track the protein down in some of the proteomics experiments necessarily. But what I would hypothesize is going on is, since SH3 domains are most often involved in kind of signaling and trafficking inside the cell, is that 873 is expressed under some kind of a stressful condition. Again, that the cell does not experience all the time, but maybe it's late in growth or very early in growth, or under some kind of nutrient starvation. And that 873 helps kind of pin a larger complex together to carry out a vital function. And that removing 873 from the genome or disrupting its structure would decrease the effectiveness of that larger complex and whatever outcome that is. So even though 873 might not itself be performing some action, have necessarily its own chemical function, it probably, I would hypothesize that it serves as a pin to hold other stuff together to more efficiently carry out a chemical reaction.
It would be, Jörg Stolke's group in Göttingen has a websites for Mycoplasma pneumoniae, JCDI, the minimal cell, and Bacillus subtilis. And they have interactome studies for Bacillus subtilis and Mycoplasma pneumoniae. It would be interesting to look and see if the orthologs of 873 in those organisms, if they have any knowledge about the interactomes of those. Maybe that might give us a clue as to why we don't see it in the proteome. There may be something that pulls it out and makes it hard to find. Don't know.
Yeah, you're absolutely correct. I can definitely reach out to them and start a dialogue and see what might be going
on. Yeah, that would be great. Other questions?
Stephen, this is Jack Dobbins. Quick question: where are you getting the protein from? I mean, are you?
Oh, we had the gene. We had the gene synthesized, and we just express it recombinantly in E. coli.
You don't have any idea whether it's on the membrane or whether it's floating in the cytoplasm, or?
I would imagine it's in the cytoplasm because I'm purifying it out of a soluble fraction. I don't have to do any membrane extraction, any detergent, anything with detergents. Based on the structure, I also wouldn't expect it to be membrane bound or integrated into the membrane at all. And there's no secretory tag, so I don't believe that it would be secreted, but that could be potentially another reason that it hasn't been identified in proteomic scans, if it's secreted directly from the SYN3 cell and is doing something else outside the cell, but that would remain to be seen at this point.
Well, to my knowledge, we hadn't picked it up
okay.
in our studies, so there's a good chance it is in the cytoplasm. I look forward to your C13 work when you get it done.
Yeah, thank you. Thanks for the question.
You betcha.